I have to assume the position for you to see me. I feel like Philip now. This is why he had his presence. All right, so I'm so grateful that people have come from far and wide to tribute our Philip. The tributes will all be given by Philip's scientific colleagues and his mentees, and we hope that each of the speakers will give us, in addition to their wonderful science, some personal recollections. So for those of you who don't know me, I'm Gloria Coruzzi, and today I speak to you from the privileged position that I had at Philip's side for more than 20 years. First at Rockefeller University, and then as a faculty colleague here at NYU. I have to get this. Where we were recruited by another visionary, Philip Fermansky, who is here with us today. He enabled us to build and share the first open labs where we embarked on our studies of Arabidopsis at the dawn of the genomics era. So now I'd like to tell you about Philip's remarkable path to a career in science. So Philip was a self-described accidental scientist. If Malcolm Gladwell had read an account of Philip's career, he would have been compelled to add a chapter to his book, Outliers. Outliers, the story of success, features people with unlikely paths to greatness. Outliers follows the intellectual journeys of the best and the brightest, the most famous and the most successful, and it asks, what makes high achievers different? Philip Benfey was indeed different. Philip never studied biology in high school or college. This is our Philip in high school at boarding school in Groton. I was privileged to meet his roommate from high school at the memorial at Duke a few weeks ago, Malcolm Edgerton. And Philip, he told me a bit about Philip in high school that I thought you might find interesting. So he said that Philip was super smart. Why are we not surprised? But not even though he was good at calculus, he wasn't particularly focused on science at Groton. And when he graduated, Philip spurned the usual colleges, and I think by that he means the Ivies, and he chose Hampshire, a new alternative college that expected students to direct their own studies, like at Brown, but they had to do their own farming. Maybe that's where he got the inspiration. And Malcolm said that Philip went there to write more than anything else. And in reading, there's quite a few interviews with Philip as well that I've read and also heard from him, that in order to be a good writer, you have to have life experiences. And he felt even though it was an alternative college, he actually dropped out of Hampshire to gain life experience so he could be a better writer. And so after 6 years of world travels, Philip worked as a logger in Oregon, a bricklayer in Melbourne, and a gardener in Japan, where a conference on the origin of life sparked his interest in science. But it was while working as a carpenter on the Seine in Paris that Philip found his two true and lasting loves, his wife Elizabeth and biology, a field that she encouraged him to pursue as his, quote, day job to supplement his novel writing. It turns out he excelled at both. For example, when Philip made the decision to go back to college and learn the sciences and then go on to get his PhD, he still wrote about science very eloquently. In this one quote, he said, "Science is one of the few professions in which we conspicuously try to reduce obscurity. It's all about adding new knowledge, understanding something that was previously hidden. And then he said, and there's also the creative aspect. You can have an idea in the morning and act on it that same day. A central theme of Philip's science and his life work was to understand how cells acquire their distinct identities. This spark was first ignited in his PhD studies with Phil Leader at Harvard, and Philip wrote, "I had a great graduate advisor in Philip Leader at Harvard. He was very supportive as I learned humility in the face of science." Philip, for his PhD studies, studied mast cells and our immune system, and he wondered how these cells acquired their distinct identities. But to find out the mechanisms behind it, Philip looked at a number of different systems, model systems, yeast, mice, but he decided that plants were a way to get at the mechanisms. And to do that, he joined the lab of Namhai Chua at Rockefeller University. And that's when I actually first met Philip, because not only were we in adjoining labs, we shared a common office, literally, and this was the day before cell phones. And so we heard each other speaking to our spouses and our kids and other conversations. Occasionally, we walked out to give each other privacy. But we certainly became close during those years at Rockefeller, which spanned about 10 years that we were together. But interestingly, Philip had two projects at Rock. One was some really highfalutin way to get at cell identity, and the other was to dissect, which he called boring. He was going to dissect the 35S promoter fragments using, in this case, beta-glucuronidase marker genes. And he was surprised to find that each piece of this constitutive viral promoter conferred expression of a reporter gene in different root cells of the plants. Observing this led me to an appreciation of the root as a model for developmental studies. So in starting, at one point we were both looking at jobs, specifically in the New York area, and we were comparing notes as we went along. But in starting his own lab, Philip, as he did for college, he turned down offers for faculty position, including one at an Ivy in New York City that I won't name. And instead, in consultation, Philip and I together chose NYU biology, and we came here because of the vision that Phil Furmansky, the then chair, had to build genomics and developmental genetics here at NYU, embracing the model systems across microbes, plants, and animals. Philip Furmansky is very proud, and I am too, that his recruitment of Philip, the other Philip, and myself was the front page article in the Chronicle of Higher Education. And the title of the article was "The Weed That Invigorated Plant Research." They're talking about Arabidopsis, not the weed that's populating our neighborhood right now. And the article begins, I'll just read you the first sentence. "A block from Washington Square in Lower Manhattan, where a cramped studio apartment with a bad view costs $1,000 a month." Remember, it was 1994. 25 researchers share about 3,000 square feet of newly renovated laboratory space with thousands of Arabidopsis plants. So it was here at NYU in the Silver Building, which we'll be having lunch over there, that Philip started his own lab, and we had an open shared lab that we managed quite well. But in starting his own lab, Philip decided that because plant cells don't move, he wrote, the four-dimensional problem of development, three spatial dimensions in time, is reduced to two dimensions with cell types on the radial axis and developmental stage on the longitudinal axis. In his lab, by combining both technical and conceptual leaps in the field, Philip's lab ushered in the genomic era to track down cell types using gene expression, which ultimately led to a gene expression map of the Arabidopsis root with Ken Birnbaum, who will tell you a lot more about the science that went on in Philip's lab. Philip was here for roughly 10 years, and there are a number of things that prompted his move to Duke, because Philip always had big, bold plans, and in part, he wanted to take them to another level, and in part because of the tragic events of 9/11 here in New York. Philip moved to Duke, and at Duke, and I'm sure you're going to hear more about this today, Philip added single-cell expression to the mix, giving birth to the root cell atlas. I call it Philip's magnum opus. It's a pointillist painting of every cell in the root. It's a gold standard for the plant community, which influenced all our work. And you'll hear from his student in his genomics class at Duke, Rahul Satija, how Rahul became interested in going into genomics because of Philip. And then Rahul developed a tool that was used, and he'll tell you about that more. Okay, now the most important thing, and this is getting towards the end of my presentation. I know I went over, but Philip's my friend, and he would give me permission. But the important thing about Philip's discoveries of root cell fates is that it transcends plants. This is evidenced by the fact that Philip recently gave a keynote talk and leading strand interview at the Cold Spring Harbor Stem Cell meeting just this past June 3rd, 2023. And for those of you who have not watched it, there's a YouTube video of Philip. And listening to him and his eloquence and his passion for his work, given at the height of his life struggle, Philip epitomizes the Japanese word gaman, which means enduring the seemingly unbearable with patience and dignity. For his seminal discoveries, Philip was inducted into the U.S. National Academy of Sciences in 2010. He was among the Howard Hughes investigators' first cohort of plant investigators in 2011, along with Joe Ecker, who will speak with us today. And Philip was honored, possibly most important to him, he was honored as an American Society of Plant Biologists pioneer in 2022, and this honor comes from tributes from his mentees and colleagues. And so he told me personally that that honor meant the most to him. So today, some of these colleagues and mentees are also paying tribute to our Philip. Four of those colleagues are shown here today, are shown in this picture when we celebrated Philip's 65th birthday at a Gordon conference, and he was having his celebration of that party in Duke, and so pretty much the whole audience left to go to Philip's 65th party. But Detlef Weigel did stay with me and Natasha, and we had the ending party. And so we missed Philip's party, but it was for a good reason. And so that's Natasha Raikhel who will speak today, Joe Ecker, and Detlef Weigel, and this is Julia Bailey-Serres, another dear friend of Philip's. And then Philip would also be hugely honored by the talks that are being given by his students and mentees who've gone on to run their own labs. And so today's speakers in that capacity will be Ken Birnbaum, our next speaker, Rahul Satija, Sozzani, Jas Dickinson, and Jose Dinneny. And of course, I tried to put on a slide of Philip's academic family tree, but it was way too wide. So I really suggest that you look at that. So I'm going to close on a personal note. I will miss Philip's wisdom, his vision, his level-headed perspective, his optimism, his charm, his laugh, and his larger-than-life presence. I always thought that President Obama reminded me of Philip Benfey, not the other way around. Both whip-smart, cool, collected, and greatly admired. Philip, your esprit de corps will live on in your academic family and in the many colleagues and friends like me who admired and loved you. Thank you.
So before we get started, we got some great new research talks from a lot of people who you've heard who have strong connections to Philip. But we thought we would like to take a few minutes, well, 30 minutes if we could be exact, to go over sort of why we're here and look at Philip's career and his trajectory and the things that he had done. And so I wanted to do this. There's so much to do. There's so much to talk about that it would be impossible to do in 30 minutes. And so I chose to do it in 3 chapters. And this is just sort of my idea of where things are going. Do I have the right one? And so I'm going to do this with Philip's own adages. Oh, that's important. So the things he used to say in the lab that sort of either inspired us or guided us, just to give you sort of a feel of how he ran the lab. And then I'm going to do it with some original collage work that I've come up with, which is not very good. Okay. So I wasn't taking myself seriously. All right. So pick the low-hanging fruit. Philip often said this. He was usually, sort of trying to guide us of, don't get over elaborate, just start small, stay focused. And that's what he did for his career. So here was his low-hanging fruit, as Gloria pointed out. The Arabidopsis root, every file is a different cell type. It's got this beautiful arrangement. And then another low-hanging fruit was a screen. This was, a few years after Bicoid was developed and discovered. And so developmental genetics was having a renaissance here. And so what did he look for? Another low-hanging fruit, short roots, right? I didn't think this screen was going to work, but it did, and it yielded these 2 genes, another sort of verbal low-hanging fruit, short root, and scarecrow. Okay, so that led to this observation that while in the wild type, that beautiful symmetry and intricacy that you could see, there were 2 cell types here. In both of these mutants, so there's the 2 cell types, that 2 of those cell types had turned into 1. So there was a cell type missing, and this was the infamous mutant layer. And so this was really interesting. These are Philip's teaching slides that he gave me, because this is what makes developmental biologists tingle, right? Is you have a stem cell, you have an asymmetric division that makes a daughter, and then you switch the division plane, and in this system, it makes 2 different cell files, which are 2 different cell identities. And so that decision is at the core of what makes all that beautiful pattern. And it turned out, as they elucidated in these 2 really nice papers, that short root actually controls or activates scarecrow. Scarecrow makes the division, and then short root comes around and specifies that identity. And so this was the start of a really interesting and important way in which all that pattern and the developmental patterning of the root was taking place. Then it got more interesting. So it turned out that short root wasn't where you expected. Here's scarecrow. it's in the cell type. There's the cell type that needs to make that division, to make the two columns. But it turned out that short root was not in that cell type. It was in the inner tissue, which is the stele. So this made things really interesting. So for one, in about 1885, these interesting, unique structures were found in plants. These are called plasmodesmata. They are little tunnels that go through the cell wall, and they connect two cells. And this was long thought to be a way in which plant cells could communicate with each other. And in fact, in the previous years, there had been some evidence by some people in this room that, that transcription factors could actually possibly move through these channels. So this would be a major thing, right? Because this is not the way signaling usually works. And there were still we had a lot of skeptics in the field that this was really happening, and we still lacked evidence that that actually meant something to the plant, that that would change the way development would occur in the course of development. And Philip realized that short root was a beautiful example. So I talked to Keiji Nakajima, who is the author of this paper, and I asked him this important picture, which is the protein localization of short root. Like how I thought this took you like a year, and he just corrected me, and he said, no, that was 2 years. So Philip realized when the low-hanging fruit was no longer valid. So here is the transcriptional domain of short root, and here is here's the protein domain of short root. And what you can notice is that in this endodermal layer, EN, there's no short root at the transcriptional level, but you see the protein there is really there. So they knew this was really going to be important, and so they went after this, and they got it. And so here's the more beautiful version of that, where you see the transcriptional domain. Here you see the translational fusion, and here it's going into the next cell type. So this was a major finding, a new way in which plants are signaling in order to create patterns and cell identities, and this is the stuff that development is made of. You see this really interesting phenomenon where short root is largely localized in the cytoplasm here and then moves into the nucleus. And this was part of a beautiful follow-up. I teach this every year in my development class. This is one of my favorite papers, where they figured out that short root actually, moves over. It lacks a nuclear localization signal. Short root grabs it, prevents it from moving any further, brings it into the nucleus. That induces a pair or a family of microRNAs. The microRNAs move back as a morphogen-like signal, and they pattern the specific cell types back in the stele where short root started. So this is just this beautiful example of how plants self-organize. That doesn't answer how the entire root is made, but it tells us a lot about how plants signal from cell to cell in a very plant-like, unique, interesting way in order to make these patterns. And I should add Ross Sozzani, who's here and will speak later, at about the same time showed that short root and scarecrow have another job where they induce this unique cyclin in that cell type, and they change the orientation of the division from that way to that way, and that's critical in making the cell type. And then even in January, Philip's lab had another paper showing that this process is highly dependent on the cell cycle, or that division. Okay. So yeah, I was reminiscing with Uka at the memorial in Duke the last month, and as Gloria said, Philip was the no drama Obama of biology. He was very calm. He always kept it very, very steady in the lab, but there were these cracks, that sometimes you could actually get a reaction from it. And if you brought him a cool result, you could kind of see that bubble up, and we all wanted to see that. And Uka recalled the moment that he brought Philip the information that short root and scarecrow were in the same family. This was the indication something really interesting was going on. Remember, we didn't have whole genome sequence. And he gave one of those reactions, and Uka was a little thrown back because this was not your typical Finnish way of celebrating a moment, but I think he learned to take it in stride. So this was a way, part of the excitement of like eliciting these reactions to Philip. It was very calm excitement, and then it would just kind of bubble up at times. Okay, chapter 2, how am I doing? Not bad. It's in the realm of possibilities. This was the thing. You would be at lab meeting, and there'd be a presentation, and his low voice, we'd all look to Philip, well, what do you think? And it would blurt, he would just calmly say, it's in the realm of possibility. And if you were a pessimist, this meant that, oh, he thinks I'm out on the fringe. If you were an optimist, you'd think, oh, this is possible. And Philip generally himself was an optimist and a creative person. Okay, so next chapter, it's 1999. The velour tracksuit was in fashion. Flip phones were the modern technology. We watched movies on DVD. And so Philip, at this point, I joked with him, he took a sabbatical, went into his office, read everything about genomics and systems biology, and emerged like Moses from Sinai with the Ten Commandments. He was a he was a genomics biologist, systems biologist. And he really started thinking in this early days of how we could use the power of these techniques to get at information. And he had these early ideas, which were, still prevalent today, that, especially from his experience with scarecrow and shortroot, that if you could learn all the genes that were active in a specific cell type, you could start to reverse engineer how that cell type is made. And this is, a dream for developmental biologists. His initial idea that he brought to the lab with this notion was to do about 30,000 in situ hybridizations. And for those of us who did the in situ hybridizations, we told Philip, okay, this is going to take about 100 years to do. It's especially laborious in plants. Okay, so indeed, as Gloria mentioned, the root is this beautifully transparent tissue. And not us only, but many labs started to make these beautiful GFP lines that expressed in very, very specific cells. And at that time, we had kind of the genomic equivalent of the flip phone, which is the gene chip. And we thought, God, man, if we could only, just a very naive thought, if we could only grab those cells and throw them on the gene chip, wouldn't that be really, really cool? And so there's an idea that was hatched in one morning in the lab to, hey, you can turn plant cells into individual cells. You can digest their cell walls. And hey, there's this instrument called the fluorescence activated cell sorter. You can go fetch those cells and pluck them out and get them in good enough numbers. And then we could put it on our beloved gene chip, and voila, we would have our cell type specific expression profile. That was about 2/3 of the genome at the time. Okay, so we immediately started thinking about this and drew up elaborate plans and presented them to the lab with due artistry. And the original presentation was something like this. And the lab didn't take it too seriously. They were very, very skeptical. And we faced bigger problems because if you looked in the plant literature, you would see that what protoplasts are known for is dedifferentiation. And that's exactly what we were trying to avoid. And this isn't just an obscure reference, it's all over the plant literature. And so the common knowledge out there was that if you take the cell wall away from a plant, make it a protoplast, you're going to lose the identity that you're looking for, and it'll all be done. But Philip was really good at spotting the difference between dogma and knowledge. And one thing that was clear was that nobody knew the timing in which all this dedifferentiation takes place. Of course, this is a famous thing about plants. They can regenerate from a single somatic cell. But how quickly did that take? And nobody had really answered that question. And we started to have strong opinions or hunches that that wasn't all that quick. And if you acted quickly, you could get this done. So Philip teamed up with David Galbraith, the only known plant cell sorter at the time. He had a naive and hopeful postdoc go back and forth and back and forth, literally with the plants on the plane. And one time the flight attendant was crushing them in, and the whole experiment got crushed. We started to get some results. That was like, back and forth and back and forth, 2 years. We had 2 cell types. Some of it was making sense. We couldn't quite figure everything out. We submitted an NSF grant. That was rejected. We submitted my fellowship. That was rejected. We submitted another NSF grant. That was rejected. But Philip, he was really good. He could take rejection. It just rolled off his back. It helps to have a lot of confidence, which he did, and good ideas. Okay, but then, so I think at this point, maybe it's 2 or 3 years in, and I go to Philip and say, what should we do? Should we just do more in situs and try to figure this out, or we take another approach? And his response was, do more cells. That was the thing that he was really good at. He knew when to just press on and when more information will help you figure out what's happening. And so that's what we did. Now, at this time, we got to 5 different cells. We eventually added stages. This is the first version. It's gotten much more elaborate now. And so what we realized is that when we got this far, we had been making mistakes about what was cell type specificity, in that we were sort of fooled by all our markers and thinking that a lot of the genome was going to be cell type specific. But in fact, a lot of it is just enriched in one area and one tissue, and also expressed in another. So when we were doing these in situs, we were picking these genes that were alternately expressed, but they were really expressed everywhere. And that was the problem. But when we did the 5 tissues, we can spot genes like this that were specifically expressed in a tissue. And so that's some of the results that came out. I remember this gene very well. We said, OK, let's try to do the in situ and see what happens. And boom, it was right where it's supposed to be. And this is my moment with Philip. I never forgot. I remember I grabbed the histogram. I grabbed the printout of this image. I went into his little office on the 7th floor in the building. And I was waiting for my response, for my moment. Was he going to curse me out? Was he going to jump up? And he's looking at this micrograph, and he's just staring at it. And then this long hand just comes up as a congratulatory shake. And I will never, I think I remember the microscope I looked at, or the room where I saw this, and got excited. But I will never forget all the details of that room and that slow hand coming up for the shake. It was another moment of excitement, and so like one of the things that really inspired me. Okay, and then if there's any more skeptics out there, I'll just go through. We did a lot of different genes. Here's SCL3 exactly in the right place. PIN2 was good. PIN4, these were a lot of markers that were there. SUC2 and GL2. And then finally we said, all right, we're ready. We can now submit this. We think we have a big story. Submitted to Nature, rejected. Philip did not bat an eye. He didn't, he wasn't offended. He just said, well, let's rejigger this a little bit. We submit it to Science, and then that's our route map. And so this was a really important part of bringing developmental biology into the genomics era in plants. It was a very important jumping off point for Philip's lab in really exploring a lot of interesting root biology, which I'm not going to get into. It was a really important resource for the community, and this is one of Philip's legacies. And as Gloria mentioned, it kept going with single cell and other techniques, but it was really Philip's confidence and vision that led to this accomplishment. And I should add, some of the things that happened after this, for example, Jose Dinneny, who will speak later today, used this technique to explore one of the fascinating parts of plant biology, which is the plasticity of cells, right? They can't move around, And so they're very, very sensitive to the environment. And Jose started measuring this as cell types respond to different stresses. And some of the big, interesting surprises of this kind of work are that different cells are responding in very, very different way and mediating the stress. And so that became a whole other area of study in the field, opened up by the Benfey lab and Jose's nice work. Okay, all right, you've seen that, right? But I got to say, Philip's the only guy who could look intimidating behind a bunch of Arabidopsis plants. Others have tried, but he is. Okay, I made the patch for this great quote, or this thing he used to say. This is Philip used to say to you in The Land of the Blind, the one-eyed man is king. I think is his version of humility. He had a lot of confidence. But also, this he used, we talked a little bit about this last night with Jazz. He really liked us, or he wanted and pushed us to strive to do things, to extend ourselves, to have confidence and do things that we necessarily didn't have confidence in doing. So if you came to him and said, should I really apply there? I'm not sure my resume is good. Should I start this company? Should I take this position? The Land of the Blind, the one-eyed man king, is the quote that would often come out. And what he was trying to do is say, you're just as qualified as anybody else. You can do this. You can do this. And so that was really important. But now I'm going to spin this to say Philip was really clear-eyed. He didn't have that one eye. And it was this really great intellect combined with a great memory that allowed him to make this next discovery in Chapter 3. All right, Chapter 3. So the lab was going into much more detail now about how the root cell identity was composed. There were much more detailed profiles of the roots. We still didn't have single cell. But now Philip was doing these slices up the root, right? So this is developmental time, and that axis, one of the things that's always really nice about the root. And so Philip was exploring this developmental time. And in this nice paper that Siobhan Brady put out, they actually elaborated on the cell type map. And then, interestingly, started looking at these slices in the developmental time. And so either intuitively or luckily, they did it on 2 individual roots, so not pooled. And here they're showing it pretty much replicates, as you would want to show, in the 2 roots. Of course, these are genes. That's the developmental series. And they showed they had things right. But they did show this one, which was an anomaly, which showed that there were some genes that were expressed one way in one root and a different way in another root. And then this is Miguel, part of Miguel Moreno Risueño's nice work. When they looked in detail, they could see that these were genes that were expressed together, and they were kind of funny patterns. And then if they looked in this next root, root 2, they were also highly correlated with each other, but expressed in a different pattern. And that went on with some other groups that you see the co-expression here, but then a completely different pattern in root 2. Sometimes oscillations, sometimes waves. Okay, and this is where Philip was really good. He had a very, very broad interest, and he read a lot of the animal field as well as the plant field. And he had a great memory. He could remember everything from every paper. And so Philip thought, and Miguel was just telling me how he sent him to go talk to Olivier Forquier, who's made these beautiful discoveries of the segmental clock in animals. This is in mouse. And Philip had a hunch that maybe his weird patterns of one root versus another might actually reflect a clock. And so what's going on here is you see this lunatic fringe wave occurring, and where it meets this determination front, it is creating a somite. And so this is how we, as vertebrates, are patterned in a lot of ways, right? And it's this kind of wave of expression, these oscillations which are elaborating the body plan during development. And so Miguel was telling me that they took this to Olivier. Olivier took a look at it, all the data, and said, you know what? I think you have a clock. And so they went back to Duke. Miguel hooked up this luciferase to one of their putative oscillators, and it turned out they had a clock. And this was a beautiful result, as it really illustrated how these two separate experiments in multicellularity have come up with a similar solution and a similar way of elaborating body plan. And so the question is, what really was it doing? And lateral roots are not embryonic, but they are the elaboration of the root body plan. And so when they looked more carefully, they saw that these oscillations, the period of the oscillations, were actually the same period at which you got a new lateral root branch origin forming. And they went on to show that the position and the origin of the lateral roots was closely tied to these oscillations. And this has also spawned a major, debates, but also a major area for developmental biologists in plants. And it led to this other beautiful paper that really documented all this. And then subsequent to that, that determination front, what's determining that the place where the sort of the wave hits the shore and you get the somite or the lateral root forming is important. And Jazz Dickinson, who's going to speak later, found some metabolites that are working in the pathway of retinoic acid, similar to the animal system, that's helping determine that determination front. So this work really went on. Philip has had a beautiful way of pursuing really the interesting results in the lab. When I asked Olivier for this that really nice picture of lunatic fringe and the segmental clock, he sends us this really nice quote of his discussions with Philip during this time. They were on a DARPA grant to find the laws of biology. That was the goal. So I don't think they totally succeeded, but actually these two guys went pretty far. Okay, and then Olivier remembers that Philip was actually also completely fluent in French, which he was. And so Olivier also remembers how Philip's vision went, that it was Philip who really started thinking about ways they could use genomics to explore the clock. And Olivier was really impressed by Philip's vision, wide vision. Okay, I'm coming to the end. I have really kind of almost scratched the surface, and I apologize to everybody whose work I haven't mentioned, but it's worth mentioning that this is actually Philip's most highly cited paper out of the lab, and I haven't even talked about it. This is where they actually lay out the ontogeny of the lateral root, and that literally started an entire new field, and it's often referred to. As we have been talking about, my original root map has now turned into an atlas, and Rachel Shahan's nice paper has really laid out the resource that Gloria was talking about. Trevor Nolan built on that and showed that in addition to cell type specific signals that you get out of single cell, you make these cell type specific perturbations that are induced at a specific time, and you can really explore the circuitry with those two powerful techniques in common. Explore the circuitry of individual cells. So in many ways, Philip's original vision with this in situs has really came to actually be realized in his lab. Okay, and then in the last minute or so here, all these are really amazing accomplishments, but one of the true accomplishments that Philip has achieved is his legacy. The lab was an incredible garden for great ideas, for inspiration. And so since that Pioneer Award, we've been trying to track the academic family tree And to date, it's sort of a work in progress. There are at least 33 independent research labs that have come out of Philip's lab, postdoc and grad students. And then we track the next generation. There are 61 independent labs that have come out of the children labs. And this will obviously grow. And sure, Philip wrote great papers. He was really clever. He had great resources. But I think one of the things that leads to this legacy is creating an environment where you inspire people and you keep their passion, and that's how you can propagate this wave of science. Okay, so with that, I leave a final picture of the big man, as we call him, who started with perhaps low-hanging fruit, but used that low-hanging fruit to show us the beautiful complexity of how plant development works in many ways. Okay, thank you.
All right, thank you so much, Ken, and to Gloria and the organizers for having me. I do want to start and talk a little bit about my time at Duke. It's really been fascinating for me, both so far today and throughout the day, to learn more about Phil Benfey and the impact that he has and he's had. I am, as an understatement, not a plant biologist, as you know. But also, I'm not actually, in contrast to Phil and also to Ken, apparently, I'm not someone who found biology sort of accidentally. I actually had a pretty linear trajectory. I left undergraduate very focused on computational and biology and genomics, and when I got my PhD and then my postdoc and came here. But the reason for that is not as an over-exaggeration, is Phil Benfey. And I'm really grateful to be here today as a representative and an example of the impact that he had as a teacher. It was my junior year in college. I guess that was 2004, 2005, when I signed up for what would have been a graduate class, but also, fortunately for me, to accept as undergraduates, just called Genomics. I don't think it'll surprise anybody in this room that Phil was an exceptional teacher. This was a class that you I don't even think of as like an undergraduate. It was something I looked forward to going to. I didn't think of it as work. It was so exciting to go every time. And I know all of us have had classes like that, and all of us have had teachers like that. And the course was called Genomics, but really it was about functional genomics. That was what he was really excited about, and that's what he wanted to teach us about. And to me, it was so incredible, and you'll see this in my work, to learn about all these different things that we can measure at genome scale resolution. That was what was so exciting, and it really came out of Ken's talk as well. And I remember most specifically, well, I remember two things. One is a question on the final exam, which I'll tell you at the end. And I've told this story before. But the other is he really walked us through. I was really interested in gene regulation and this idea of being able to detect transcription factor binding. And he walked us through the CHIP assay, chromatin immunoprecipitation, and all the different steps and how long that would take to be able to detect these binding events. And then he said, we used to do this with qPCR, but now we can look across the entire genome with ChIP. Or with ChIP. That was the technology that really got me excited. And actually, that whole course, there really wasn't very much sequencing involved because in functional genomics, it was mostly microarrays. But I think that, and I say this to my students now, that was the moment where I think the field, moving from microarrays to sequencing was exciting, but it was really the ability to look genome-wide with arrays, as Ken showed so beautifully, that really sort of opened my eyes and was so exciting for the field. And this idea that, we can measure all of these different things across the entire genome in a single experiment. As someone who was interested in computational biology and a computer scientist, I thought, okay, this is where it's at. And it really sort of confirmed and set me down the path. And Phil and others at Duke, including Greg Ray and Alex Hardemink, are very much the reason that I'm here. I fortunately stayed in touch with Phil even after leaving Duke. When I was concluding my postdoc and looking for jobs, I also actually was choosing between NYU and another Ivy League university in New York. And I emailed Phil. I hadn't talked to him in a while, and I just said, I love your thoughts and advice, and what do you think? And I've told this story before, and he said, I think you should go to NYU. And then he said, I think NYU is a really fantastic, incredible place to open a lab. I think it was being slightly tongue-in-cheek in doing that, or maybe he said for the first stage of your career. But I reconnected with him then and just talked generally about sort of challenges and and opportunities for junior faculty, and it's pretty special, maybe unsurprising, pretty special that he would take that time, for someone just that had been in one of his classes. I should also mention, there's another genomic pioneer, his name is Amit Khera, who's a PI at the Broad and started his own cardiovascular disease company that was also in that class, in the same room, which is pretty amazing. And then the last thing that I'll say is the last time I saw Phil is I went back to Duke. The students, including people in his lab, invited me to give a talk just before the pandemic. And it was the first time I met with him on my visit. It was the first time I actually talked about research with him. As an undergraduate, you never really think about the incredible impact that your teachers have. And we talked about single cell, and it was incredible to me. And he told me about the root cell atlas that he was building, and I met Rachel Shahan in his lab and saw kind of that early work that Gloria referenced at the beginning. And you'll see from my talk today the value of building references and mapping data to them. And one of the things that Phil did was he used Seurat to build that root cell atlas from healthy plants and then mapped mutant cells onto those developmental trajectories using some of the algorithms that we built for reference mapping. I can't tell you what a thrill that was for me. And for those of you who are more junior in the audience, I think if you ever get that opportunity for the people who kind of got you started in the field to come back and then watch them benefit in their own science and labs for the things that you had built because of them, it's pretty great.
Thank you for inviting me to help celebrate Philip Benfey's life and work. From what we've heard so far, I got the impression that Philip was somehow a cross between a saint and a prince. And I did look for evidence of Saint Prince, of Saint Philip. Couldn't find evidence of Saint Philip, but fortunately my photographic evidence was given away a little bit already, but I wanted to share with you this photographic evidence of Prince Philip. I think it captures him really well. Fortunately, he was a prince who didn't take himself too seriously. I first got to meet Philip actually a little bit over 30 years ago when he had just arrived here together with Gloria at NYU, and I had heard that the two of us were sharing this interest in transcription factors and development, and I was still a postdoc. I cheekily invited myself to NYU, and he was really extremely gracious that he said, yes, come visit, and that he entertained me with what he was doing. I think really this reflects his spirit of generosity that we've heard about today. And it was really an eye-opener for me to, I as a reformed Drosophila geneticist, to hear that there was actually stuff going on underground, and that was pretty important and interesting stuff. Philip and I, we've not really formally collaborated a whole lot, but this initial interaction led actually to a joint grant application. We heard about this from Ken already, to look at the expression of all transcription factor genes in both the Arabidopsis shoot and the root. And there were these two parts, and so perhaps the more conservative European mind-numbing part was those a very large number of in situs that we're going to do in the shoot and the root, and this other creative Phil Benfey kind of part was to use to combine the cell sorting with microarrays. And so not only did that win the day, perhaps at least in part because the original grant application was turned down, so maybe we would have been sucked into doing all these in situ. So we had already heard this, that Philip, he would just shrug and walk on and see the good actually in that happened. And this was really, groundbreaking, revolutionizing for developmental biology. And we've heard a couple of times about this paper, but I think there is another teaching point that hasn't been mentioned. Ideas are cheap. And in the end, actually, you have to act on these ideas. And so there had actually been an early paper on combining cell sorting with microarrays in Drosophila. Hannela Ruhala-Becker had published a paper in '99, but the field had not followed up on it. And Philip actually recognized how transformative this technology would be. And he didn't just profile one tissue, because what you really learn from this. But again, as Ken told us, 5 different tissues, 3 different stages, so 15 different profiles to really show this. And this technology then launched the career of many of his postdocs, Ken, Jose, who will talk later, Siobhan, and so on and so forth. And then I want to highlight one other paper that is perhaps my personal favorite of all the papers that Philip has published, because this paper here, and it's built on an initial discovery that Kim made, who is here in the audience, is actually a very different way how plants have solved a signaling problem. So we've heard about the mRNA of short root being made in some cells, and then the protein moving to the next cell. But for me, this is really one of the coolest results ever, how it induces another transcription factor that then dimerizes, sucks it into the nucleus, and can't move any further. And it's really absolutely beautiful piece of biology. All right, so that is what I wanted to say about Philip. He was a giant in the field in many different ways, and I feel myself being very fortunate to have come to know him at least a little bit.
But what I wanted to put out here is what we heard this morning in terms of the single cell, but also to put it in the vision that Philip had. And I'm going to touch just a couple of minutes on the 3 words here: mentor. It was my mentor, and as a lot of us, it was always at the other line of the phone when you needed, and he would always respond, and he would always advise within less than 24 hours. And as a mentor, I remember when it was April of 2022, and I got my letter that I was promoted to full professor. And then I read the dean letter that supported that, and I called him up, and he would pick up immediately the phone, and I said, "Philip, my dean wrote, Dr. Sozzani is the whole enchilada." and then I heard this beautiful laugh of Philip that just echoed and said, "That's a good thing, Ross. You're the whole enchilada." And then immediately he said, "Great, so now you're a full professor. Let's take away the barrier of, and let's work together, because you don't have any more to prove that you're independent or whatsoever." And so that was May when Philip brought together all of the university in North Carolina, all of the community college of North Carolina, and led us to a vision that is now implemented. So it was there in September when we had the phone call from the program director that we were awarded an NSF engine to build a North Carolina Ag Innovation Corridor. And so what that meant was that Philip, as a writer, wrote these 2 beautiful pages about the scam benefit of the RTP and how the Duke, the NC State, and the UNC, and all of the biotechnology over there had actually take away aspect to the rural North Carolina, and how for this 2-page and reference to Jim Crow, he really wanted to leave an innovation ecosystem in North Carolina in ag, saying, "I have been a plant biologist for so many years, and now I want to give it back to the community of the people that need the most." And so he led the engine that is called CROPS, and he was on the phone call when the program director said, "We're going to award you, and we want you to continue in that space." And so the entrepreneurship also comes along with engine come with 4 important aspects: workforce development, ecosystem development, user-inspired research, and translation to practice. And so translation to practice was part of Philip's entrepreneurial, I would say, dynamic behavior. And as we were discussing yesterday, and Detlef said that, Philip was a man of, he would have the idea in the morning, and by the evening, it would be implemented. And so we were talking about the success that he had in the lab for the single cell and the atlas, and he would complement on some of the aspects in terms of the AI model that we were developing, both his lab and my group, and then other technology. And then he looked at me and says, how about we found a company? And then I look at him and said, sure. The day after, I got the retainer from the lawyer, and we both paid for that, and I was the co-founder and president of Raleigh Bioscience, and Philip named Raleigh Bioscience because he wanted to be launched out of NC State and the new plant science initiative that he helped to build in terms of ecosystem for new startup to have the support. And then he said, let's give a great name like Sir Raleigh Walter. And so that's why Raleigh Bioscience. We are housed in the Plant Science Initiative. We are in Raleigh, and you will see later on that there's another story associated with it. And then at the end, I'll tell you in terms of why it was a visionary, developmental genomics, and how he would put himself in a room full of people, not in academia, but in industry, and tell one thing that they did wrong.
Thank you so much to the organizers. I'm really honored to be here as one of Philip's former mentees because I know Philip took a lot of pride in his mentorship, and his record was astounding in terms of mentorship. Ken mentioned earlier there are 33 independent labs that came from Philip, and that's just looking at Philip's former postdocs. Philip also mentored many postdocs who have been very successful in industry and in other diverse careers. Philip mentored graduate students and undergraduates. So there's this amazing community of people who were mentored by Philip, and so many have been so successful. So to be representing them is a major honor, and I just want to acknowledge that whatever stories I tell, I think, could easily be amplified, and there are so many more stories that we don't get to hear today. And I thought I would start by talking about how I learned about Philip. So my background is actually in chemistry and kind of biomedical engineering. I did my graduate work at UNC in a lab where I developed technology to look at the chemistry of leukemia, or the small molecules that are affecting cell behavior during cancer. And what we would do is we would take cells, I would put a fluorescent substrate in those cells, and I would use what was basically a microfluidic device to see what happened to that substrate for enzymatic activity. And there was a lot of interesting chemistry happening, and that made me really curious about what was happening in these cells. And I became really interested particularly in leukemic stem cells, which are thought to sort of give rise to leukemia and also maybe be the cause of very highly resistant cancers, leukemic cancers. And as I was doing all this chemistry, technology development, I just became so curious about what makes a cell divide or change its shape or change its function, what makes it differentiate. And I could see in my data that there was lots of really interesting chemistry going on in these cells, but this was a pretty hard system to study that developmental process because leukemic cells are in a big mass. Leukemic cells are in a big mass. You have to separate them from the blood, and then you have no idea which are the stem cells or not. You have to try to narrow that down, try to find a population which you think are enriched with stem cells. It was very, very challenging and kind of unsatisfying. And then I was talking to my husband, who was at Duke University, and he said, oh, this postdoc named Sozzani came into my class today, and she was talking about this really cool model system that she studies in Philip Benfey's lab. and I was thinking about where to do my postdoc, and he said, this could be really cool. This is a really cool system. And as has also been talked today, what's really exciting about this system is it's 2D. It takes a 4-dimensional problem and it simplifies it into a 2-dimensional problem. And this is something you can look at a single time point and get so much information about, exactly which cells are the stem cells. You know exactly their history, basically, of their behavior, and can make a lot of interesting hypotheses and discoveries based on that. So one thing that, and another thing that Philip had done was he had actually published microfluidic papers, or a paper on microfluidics. So we had actually reviewed that paper in my lab as we were writing a review on microfluidic devices, and before I knew anything about Philip, we had talked about how amazing this paper was. Because so many microfluidic technology kinds of papers, they design something and then they figure out what biological process might be interesting to look at with that device or that technology. But Philip had a biological problem, and he saw technology that could address that problem, and then he used it to measure a real biological question. And we reviewed hundreds of papers, and there were only a handful that did this. And so this was really a standout paper. So I was really excited to hear my husband talk about how interesting this system was. And then another thing that I just want to point out is that what Ross was talking about, Philip really cared about how he could impact society, how this work that he was doing could lead to an impact in society. And I learned a lot from Philip about how to think about that and how to start to approach that. So I want to say thank you, Philip. Without Philip, I would not be a plant biologist, and I would not be working towards these really important issues of climate change and agriculture. And I wanted to reference the Prince Philip photograph that Detlef showed, because I think we are all talking so positively about Philip today, and if you didn't know Philip or know us, you might think, oh, they're just talking about that because it's a special memorial ceremony. But this is actually how we always talked about Philip. And my husband would make fun of me and the people in the lab that I would talk with, because he would say we acted like he was a prince or a king. That's how much reverence we had when we spoke of Philip. We always spoke of him so highly. And he was like, you're not supposed to like your boss that much. You're supposed to complain about your boss sometimes. And we really didn't. And for many years, as I've started at UCSD, or the 4 years as I've been at UCSD, I think a lot about, well, why did we like to work with Philip that much? Why was he such a good mentor? And I think one of the things that has come up today is that he was just a source of so many pearls of wisdom. The low-hanging fruit, knowing when to go after low-hanging fruit. We've talked a lot about today how he was very resilient to rejection. And one thing he would tell us is, you're going to get rejected a lot in academia, and what you can do is you take one day to feel really sorry for yourself when you get that news, and then the next day you brush it off. So give yourself time to process, but then the next day you tell yourself it's time to move on and you move forward. He also said, told me ideas are cheap, which I think Detlef also mentioned. It's the work that's important. So don't get freaked out when somebody has a similar idea to you. That means it's a good idea probably. Just do the work, and that's what's the hard part going to be. Philip also was really good at teaching you to know your own worth. He would tell you, don't be afraid to walk away if somebody is giving you a bad offer, if somebody is not valuing your time. You don't have to put up with that. You should know that you are just as important as they are, and you should carry that forward in your interactions. And that kind of brings me to, I think, for me, what was the most powerful thing about working with Philip was that he was such a strong advocate for the people that worked for him. And he had your back. And Ross mentioned, you could call him even after you left his lab. You could call him and ask him for advice, and he was always so happy to give it. And I'm getting emotional because we actually had a patent together, and about 2 weeks before he died, we had a meeting about this patent that was scheduled with a company, and we were going to try to get the company to be interested in our patent. And Philip attended that meeting even though he was feeling very sick. And he attended that meeting just to advocate for me as the person that this company should be interacting with. And and then he left the meeting once he had done that. He said, I'm here to tell you that Jazz is great and you should work with her. And that's just one, small fraction of the advocacy he did for me. And then think about all the people he did that for. He was so kind and generous with his time and with his support. And I'm really, really grateful for that.
I want to reiterate what Jazz said, that it's a tremendous honor to have this chance to be able to represent other mentees that have had the luck and opportunity to work with Philip. I chose this title essentially thinking about Philip. Of course, I think he first thinks of himself as a developmental biologist, but he also has a sense of adventure. And that was really one of the main reasons that I was so attracted to his lab, was that he had great taste. He chose a great system, the root, and I'm really happy to have transitioned into that system myself. And he also had a really great sense of technology, that he could see things on the horizon before anyone else did, and invested in things to a large extent that played out very well. There was, I think, some imaging using styrofoam at some points that didn't play out so well, but most things were very, very well chosen. Another aspect, so I was in the lab for a fairly short period of time, but I was really, I must say, taken aback by his understanding of how to mentor people in the lab. And he gave you, in some ways, what you needed. And sometimes I didn't know what I needed myself, but he provided that support. So my family background's a little complicated, to say the least, but my parents were also adventurous, perhaps too adventurous. And when I came to learn about Philip's background, he also had that period of wanderlust and roaming about the world. But yet he was also responsible, right? And he came back, he did what he needed to do, he supported his family, and he raised the lab in a very responsible way. And yeah, I really appreciate that. And one time he took me to a basketball game with his kids. So Duke has this team that's so, it's the students that are really sort of invigorated when they're watching this game. And the funny thing was, it was like it was like a minute before the end of the fourth quarter, and the Duke team catches a rebound, but they're confused, and they actually go to the opponent's hoop and shoot the ball and get it in, and that changes then who's going to win. So there's seconds left in the game, and so then finally Duke gets the ball and then hits it into their goal or dunks it, and it was just such a joy to be there. He took me with his sons, and it was the type of experience that I hadn't had before. So thank you.
Thank you. Thank you, Gloria. Actually, I will probably not talk at all about plants today because I decided that I usually go over time by twofold when I do that. And my students are going to be speaking, my students, meaning actually they're all postdocs, I call them students, they're all speaking at many meetings this summer. So you'll actually hear it from the horse's mouth instead of from me. Now, where is the pointer? Okay, all right. Okay, so this is something that Philip and I had in common. This idea that some new method developed can drive new biology, and that's really a bottleneck. And Philip was really in that space. He and I saw that, very, very similarly that you needed the next microscope that was better, sequencer that was better, approach that was better, root array or chip array that was better. I've seen a few pictures of Philip, but during his 65th birthday, I sort of made up a little story because he's sort of a superhero. And so I cartoonized him to this. And the two photos that I've seen, one from, I don't know if it was from Detlef with the prince, and here he's got his fist ready. He was excited about the science. And so that's what I always liked to talk to Philip about, like what was the latest thing that he was thinking about. And he would tell me even things that he was planning for years to come, right? This is where I want to go, right? He was not shy about saying what he was going to do. He wasn't worried about that. He knew that, you could have a lot of ideas, as Detlef said, but you have to drive those ideas home. And so Philip would get very passionate. We had a lot of opportunity to talk to one another as we served for many years on Magnus Norberg, a friend and colleague, and who's director at the Gregor Mendel Institute. And so we would once a year spend a number of days together and discuss science. So I really miss him. In fact, Philip gave me financial advice as well. I'd call him and say, hey, where are you investing your, when we became Hughes investigators, you got this like extra money. And he was like, what are you going to do with this money? He'd say, well, here's a list of things that I'm investing in. I said, okay, thank you, Philip. So he was even providing financial advice. To this day, those things have outperformed what I picked. So I really want to thank you. And the community is recognizing Phillips. I mean, these are your peers here. The North American Rapid Steering Committee has basically honored Philip by calling this sort of Lifetime Achievement Award, the Philip and Benfey Rapid Opposite Community Lifetime Award. So that's really an honor that I think is going to go. Yeah.
Thank you, Gloria, and thank you so much for organizing this meeting. It's absolutely beautiful science. It's very hard to speak after Joe, of course, for me, but nevertheless, it will not be very scientific talk. But I have to say that many of us probably will be agree that during this meeting, symposium, we all learn so much more about Philip than we knew before. It's amazing how many new characteristics, his new Philips we've learned. He was an incredible, incredible man. As I reflect on Philip's remarkable life and legacy, I'm thinking about the profound impact he had on who had the privilege of knowing him. His dedication to his work and his passion for scientific inquiry were truly remarkable. But it was his kindness and compassion and ability to connect with people on a deep level that set him apart. As you have heard during this symposium, throughout his career, Philip made groundbreaking contributions that have shaped our understanding of plant biology. His innovative research paved the way for countless discoveries and advancements in the field, and his mentorship of students and postdocs left an indelible mark on the next generation of scientists. As a colleague, Philip was not only innovative and bold, but also incredibly supportive. His insights and suggestions as an associate editor of Plant Physiology when I was editor-in-chief was invaluable. And his dedication and excellence played a significant role in elevation of this journal stature. His commitment to scientific integrity and his tireless work ethics set a high standard for us all. During the last 10 years, I worked with Philip on the editorial board of PNS and the National Academy of Science Journal. Philip always handled a large load of manuscripts and never asked for a break. Even now, 8 months after his passing, I'm still getting the manuscript that were originally handled by Philip to make a final decision on their publication. This is some of us during National Academy meeting. Philip's leadership of our National Academy Plant Biology section was exemplary, and his ability to communicate his idea and opinions with clarity and conviction made him a very respected voice in our field. The way Philip talked and delivered his ideas and opinions was impossible to ignore, and people usually followed his suggestions. On the other hand, he was a very attentive listener and open for discussion and changing his views. He was also very effective in representing our sections in numerous National Academy of Science activity, especially a section that responsible for the last section represented all representatives from all fields of academy and deciding who will be elected or not. He was so convincing and so effective. It was very special. Since I'm the last speaker, I would like to share with you my memories of Philip as a colleague and friend. I knew Philip as a colleague and worked with him for many years, but I developed a very special relationship with Philip and with his wife Elizabeth when approximately 20 years ago, they started to come to Pasadena, California, once or twice a year at the place where I live. Philip was a kind and compassionate friend. Our conversations were always stimulating, filled with laughter, and enriched by his vast knowledge and curiosity about the world. He was a ferocious reader and had a unique ability to connect with people on a deep level. I have heard this over and over again from my own students and postdocs who had a chance to discuss their science with Philip. They were truly amazed that he was seriously listening and engaged with them, discussing their research that was not directly in his area of interest. One of the things that I value most about Philip was his love for culture and arts. Our outings at Huntington Gardens, museums in Pasadena and Los Angeles, and concerts at Disney World Concert Hall were always highlights and special moments in the time spent with him and Elizabeth. His appreciation for beauty and creativity was infectious, and it left a lasting impression on me. I'd like to share with you a memorable experience we had attending a concert at Disney Hall featuring a renowned violinist, Joshua Bell. The performance was nothing short of spectacular, as one would expect from someone of Bell's extraordinary talent. What made the evening even more unforgettable was the surprise encounter we had during dinner afterwards. The fate would have it, our table was situated next to Joshua Bell and two of his friends. The excitement and joy on Philip's face were palpable. He was like a child on a Christmas morning, thrilled to be in such close proximity to the celebrated violinist. It was a moment of pure delight and testament to Philip's love of the arts and the joy they brought into his life. On this slide, I'd like to take you back to a cherished memory we shared together at a symphony concert held at the open air at Huntington Garden in Pasadena. It was a delightful evening filled with music, laughter, and warm friendship. The concert was preceded by a picnic dinner, and as you can see, we gathered around the tables, each bringing our own culinary creations. One of my friends, known for his culinary prowess, prepared a stunning dish called torta rustica that stole the show. It was a dish so exquisite that passerby couldn't resist capturing its beauty in photographs. I recall Philip's genuine delight as he savored each bite. His curiosity piqued as he inquired about the recipe and ingredients. Oops. And here we are captured in a moment after a delightful dinner at one of our favorite restaurants in Pasadena, and an establishment that boasted a magnificent selection of cheeses, Philip's weakness. He would often select cheeses to take home, to his hotel, declaring them unmatching in quality and flavor. But Philip was, of course, more than just a connoisseur of the arts and great food. Oops. What's a minute? No, it's, oh. He was a devoted husband to Elizabeth and loving father to his sons, Sam and Julian. Philip's relationship with Elizabeth was a true testament to the power of love and partnership. Their bond was palpable, and it was clear that they brought out the best in each other. He adored Elizabeth with unwavering love. And what's the matter? On this slide, we have a glimpse of Elizabeth and Philip at the National Academy Gala. Their presence illuminated the event, radiating warmth, elegance, and joy. As a couple, they were truly remarkable, graceful on the dance floor, engaging in conversation, and sharing laughter with everyone around them. Their genuine happiness and affection for each other enriched all who were fortunate enough to be with them. This picture was taken during my retirement party, holds a special place in my heart. Despite his busy schedule, I was honored that Philip made the journey to California just to join my celebration. His presence was a testament to the depth of our friendship and the kindness of his heart, a gesture that touched me deeply and underscored the generous nature of this man we knew and loved. You can see Gloria there too. You have seen already this slide. Gloria started when she gave her talk at the beginning of this wonderful symposium. As Gloria said, this was a snapshot of us celebrating Philip's 65th birthday during the Gordon conferences that Gloria organized. Among the familiar faces, you of course recognize some of the speakers from today's symposium. It was a joyous occasion marked by the laughter, camaraderie, and the shared appreciation of Philip's presence in our life. Here we see Philip surrounded by his beloved family, Elizabeth, Sam, and Julian. This precious moment captured in August 2023, just a month before Philip's passing. And it serves as a poignant reminder of the bond they shared. Even in his absence, Philip's legacy continues to inspire and influence us. And the talks by his former laboratory members today are a testament to that. His voice may be silent, but its impact will be felt for generations to come. In closing, I'm grateful for the privilege of knowing Philip as a colleague and friend. I will always remember and cherish his brilliance, kindness, and passion for life. As the 13th century Persian poet Rumi once said, "Goodbyes are only for those who love with their eyes, because for those who love with their heart and soul, there is no such thing as a separation." Philip's legacy lives on in all of us. Thank you.
So Natasha's remarks are remarkable in expressing how much we've lost, but let's take her advice to know that his memory will not only be with us, but his Philipisms, his mentees, and he would not want us to be crying, even though we are. And let's just try to keep his spirit alive in those aphorisms and keep the bonds with each other. So I want to, before I close though, I want to, obviously, Elizabeth, several of us here were at the Duke Memorial celebration of his life, as Elizabeth would say, and she forbade us to wear black, even though for some of us that's our costume. And it was beautiful. Stories. Philip's cello was played by an expert musician, playing pieces that he was working on at home. And so I feel like Elizabeth is with us and her boys, and I have to say that Sam has Philip's mannerisms, and he's from the back. I thought I glimpsed Philip a few times. And Sam's wife's family is here to represent the Benfeys, and I just want to thank Aiden Mortman, her sister, and Hila Arnan, her mother, Sam's mother. Did I get that right? Sam's wife's mother, that's what I meant. And so they are representing the extended Benfey family. And I know that Elizabeth is very grateful that you did that for the Benfeys. So thank you from us. I'd also like to thank the NYU biology staff that made this symposium possible, both in the planning and the day. Tara Rock, the Director of Administration, and her terrific team, Yasmin, Ashley, and my assistant, Samantha, who's helped enormously. And I'd like to thank John and Louis for their help in the AV. And I'd like to thank NYU Campus Media for their work on the production and services. This symposium is being professionally recorded and will be available. I'd also like to thank Ken in absentia, both for organizing the meeting and the speakers and for remembering to go to his daughter's graduation. Absent-minded professor, I guess. And I really want to thank everybody for coming, including some NYU biology alumni, and especially the other Philip Furmansky, who really graced us with his presence. And without Philip Furmansky, we would not have had Philip Benfey here at NYU. So I'd like to have a little thanks for Phil Furmansky. Actually, we wouldn't have many of us in this room. Steve Small, Justin Blau, who else? Me. But truly, Philip Furmansky hired about 14 faculty, and I had the tree of life of all his hires. And Philip Benfey and I truly came here because of Philip Furmansky's vision to build a department. And so, in closing, if your name is Philip, and actually Philip's mentor was Philip Leader, so it's Philip Leader, Philip Benfey, Phil Furmansky. All right, so thank you all for coming. We have a reception that we'd like you to join, and thank you.